Hi, my name is Ashlyn,
And I'm Natalie.
and this is our webinar on amylase.
So, a little bit about amylase. Amylase was actually the first enzyme to be discovered and isolated in 1833. It's responsible for breaking down polysaccharides into monosaccharides, and it's produced by the salivary and pancreatic glands, while it's also contained in certain food. The breaking down of amylase into monosaccharides is actually the body's first chemical mechanism of digestion. Amylase is encoded by AMY1A or AMY2A, depending on the location of production. So AMY1A produces amylase for the salivary glands and AMY2A for the pancreatic glands. Diets low or high in starch actually relate to the number of genes of copies of genes that are spliced. And another interesting fact is a mutation in AMY2A can increase the risk of type 2 diabetes because it disables the production of pancreatic alpha amylase, which we'll talk about more later. Amylase belongs to the hydrolases class of enzymes because it catalyzes hydrolysis, which uses water to break down the bonds in larger molecules into smaller molecules. There are three types of amylase: alpha, beta, and gamma. Alpha amylase is the most common, cleaving the internal glycosidic bonds in starch. Beta amylase is specifically found in plants and some bacteria and fungi, while absent in animal cells. And gamma amylase is similar to alpha amylase, but it's found in acidic environments and it breaks the glycosidic bonds of the non-reducing ends of the sugar. Like most enzymes, amylase is named after what it cleaves, so amylose, which is the unbranched homopolysaccharide linked by alpha 1 to 4 glucose monomers, which is found in starch. Amylase randomly cleaves the interior glycosidic bonds through covalent catalysis. Here is a mechanism of amylase. So in red here is the amylase enzyme, and the yellow hexagon chain is your polysaccharide. It binds to the monomers, bind to into the active site to produce the short chains of glucose called maltose. A little bit more about alpha amylase. It's the most common type. It's found in humans, plants, and bacteria. The structure includes three domains: A, B, and C. Domain A is the largest portion of alpha amylase. It's where the active site is located, which the active site is determined by the aspartic acid, glutamic acid, aspartic acid sequence. Domain B is the calcium binding domain, where calcium ions stabilize the structure of the active site. And domain C, which is the C terminal beta sheet domain. The medical field measures the activity of alpha amylase quite a lot. It's very productive in becoming a diagnostic tool for pancreatic diseases and understanding an immune response. Because as you get sick, your saliva production increases, and since amylase is within your saliva, it's the body's first defense of trying to keep that pathogen out. Here is a color-coded of the domains of a monomer of alpha amylase made through the protein bank in Europe. In blue here is domain C. As you can see, there's a bunch of beta sheets. Domain A, the largest portion of alpha amylase, and this beta barrel, which is another defining feature, which I will talk about later. And green is domain B. So here's another picture showing the secondary structures of alpha amylase. In green is the beta sheets, and this grayish blue are the alpha helices. Here you can see this is domain C because of all of the beta sheets. It's very easy to point out. And here is kind of like an inside shot of that same picture, but it's showing the beta barrel, which is made up of 8 beta sheets right here, and surrounding it on the outside are these alpha helices.
The study we wanted to focus on was the examination of active sites of human salivary alpha amylase, or HSA, by Kandra and Gaimont. This study focuses on investigating the active sites of human salivary alpha amylase to better understand the binding regions and catalytic activity. They use substrates, malto-oligosaccharides modified by chromogenic and benzylidine groups. These molecules are considered glycosides, which are molecules with a sugar portion or the glycone region and a non-sugar portion or the aglycone region. The chromogenic region or the chromogenic group generates a measurable color change upon enzymatic reactions like cleavage. This group was treated as analogs of glucose residues in the glycone region. The benzylidine group is used as a protecting group, allowing the researchers to investigate the binding subsites of amylase, giving insight to the binding preferences and active site properties. This group is used to understand the hydrophilic and hydrophobic regions of the enzyme. The substrates range from 4 to 9 degrees of polymerization, which refers to the number of repeating monomeric units of a polymer molecule. These groups were used to allow the researchers to track and quantify the enzymatic activity via high-performance liquid chromatography, or HPLC, and diode array detection. HPLC was completed on an amino propyl silica column, which was used for product analysis, which enabled the researchers to understand the product patterns and cleavage frequencies. The diode array detection was used to detect the chromogenic reaction products and was set at 302 nanometers. In the picture, at the top is the benzylidine group, while at the bottom is the chromogenic group. This table is showing the action patterns and cleavage frequencies of HSA on the chromogenic malto-oligosaccharides, or CNP. These all have degrees of polymerization ranging from 4 to 9. The products are quantified as molar yields of glycosides containing the chromogenic group relative to the total number of products. Under the substrate column is the list of malto-oligosaccharides. The numbers next to the G refer to the degree of polymerization and the chromogenic group at the reducing end. Under the products column is the molar yield of cleavage products with CNP at the reducing end. Shorter substrates like the G4-CNP and G5-CNP show cleavage predominantly occurring at the non-reducing end, producing G2-CNP as the main product, which you can see is around 85 to 86%. Longer substrates like the G6-CNP and G9-CNP show a shift inwards towards the chain, with G3 and G4 being the more dominant products, showing that instead of cleaving at the reducing end, we're getting closer and closer to the non-reducing end. It should also be noted that the cleavage frequencies decrease for terminal sites as substrate length increases, so it's getting farther and farther away. This table focuses on the modified substrate with the benzylidine group at the non-reducing end. It's showing how HSA processes these substrates and quantifies the yields of benzylidine products. Similar to the last table, the substrate column lists the benzylidine blocked malto-oligosaccharides with the number next to the BNLG referring to the degree of polymerization, and the nitrophenol group at the reducing end. Under the products column is the molar yield of the cleavage products with the nitrophenol group at the reducing end. Cleavage becomes more varied as the substrate gets longer, as seen by the greater number of products in G8 compared to G4. So we're only seeing one cleavage product with G4 compared to the 4 cleavage products with G8. The yields of G2 NP decreases with substrate length for the most part. So we're seeing that when you're looking at G2, we have 83% with the G5 versus the 27% at G8. Subsite interactions suggest a favorable binding of the benzylidine group to subsites -3, but unfavorable ones to the subsite of -4. So for the results of active site configuration, they found that HSA has at least 6 subsites, 4 glycone or the sugar portion of the glycoside, and 2 aglycone or the non-sugar portions. They also saw that primary cleavage was between subsites -1 and +1, or between the glycone and the aglycone regions. Um, this often releases the maltotetrose from longer substrates, so that 4 monomer sugar. For substrate specificity, the benzylidine modified substrates suggest that subsite 4 is hydrophilic, while also comparing to the porcupine pancreatic alpha amylase, there were structural differences. For the cleavage patterns, it showed that shorter substrates cleaved at very specific points favorably, while with longer substrates, there was much more diverse cleavage patterns. Based on the results of the active site arrangement in HSA, it demonstrated that it is much longer than we had previously thought. These findings contribute to the refinement of the subsite model for depolymerizing enzymes. Understanding the enzyme specificity and action patterns can allow researchers to improve diagnostic assays for diseases involving alpha amylase like pancreatitis. Finally, by understanding the structural differences between HSA and PPA or the porcupine pancreatic alpha amylase, suggests that there are potential avenues for developing enzyme specific inhibitors or treatments. And here are our references. Thank you.
Thank you.